I'm Daniel Kirschner. I'm the Executive Director of Analytical Services and Biopharmaceutical Services at our Cambrex Durham site. I love chemistry. I think, there's so many careers I could have chosen. Chemistry was the one that really caught my interest. I love seeing new molecules every day, and so anytime I get a chance to look at a new chemical process, try to figure out the analytical controls around that process, that really just is a passion for me, trying to find new solutions. It's all like a puzzle and just trying to find ways to solve that puzzle. I worked in pharma, so I've been on both sides of the table. So I know what it's like to work and manage with CDMOs, and having sat at both sides of the table, I think our team can really provide them a great solution. We provide full CMC services throughout the life cycle of the drug, from early preclinical through commercial. We do method development, method validation, release, stability. We do impurity identification, nitrosamine testing, extractables and leachables. Cambrex has a really strong science focus and is very client-centric. So, at the Durham site, we have project teams that are very oriented towards our clients. It makes it a great place to work because it's a lot like pharma in a lot of ways. We get to work on the project from start to finish. Maybe we only have a small aspect, or maybe we have a broader aspect, but we get to develop strong relationships with our clients and hopefully help them and become key stakeholders in the development of their program. Having been a client myself, it's absolutely critical to build trust as a client. We want our science to show in the work that we do, but I think it's important to say that we also want to make sure we are key stakeholders in your program. And so for us, that's just a critical part of how we do our business. When we look at an example of that would be, we have charged aerosol detection, a universal detector that we use to look for trace impurities. And we had a client that came in with a neurosteroid that didn't have a chromophore. We started that method development project with them, but we couldn't leverage our team's multi-column screening platform because of the detector configuration. Fast forward a year later, we have 2 multi-column screening systems, all with CAD detectors, and that is kind of the principle by which we kind of have developed our programs across the site and continue to grow and expand our relationships with clients. We've seen quite a few changes in analytical development and testing. I think that, the introduction of the ICHQ 3D requirements that came out several years ago now changed how we look at elemental impurities using ICP-MS. Recently, the alkyl boronates, a new class of mutagenic impurities came up. They're used in Suzuki cross-coupling reactions, which is one of the most fundamental reactions for carbon-carbon bond formation, so impacts a lot of pharma. So we've been developing techniques for that. If we just step back a little bit and see how the regulatory environment is changing, the FDA has really started to push for robustness and analytical development to be moved into the early development and not something that's done at the back end of validation. So that is very clear in ICHQ2 and Q14 and the new draft guidelines that they really want the method to be built robust from the beginning and not to be shown to be robust at the end. Navigating that with our process chemistry team, our manufacturing team, and our analytical teams, it can be a challenge, but we have a really solid background in understanding the guidelines, so we understand ICHQ3A, B, C, and D. We can really clearly help them set their specification strategy and develop a control strategy to get them into the early phases and be able to get into first in human. We've destroyed silos. We really focus on trying to cross-train our team members to be able to handle an impurity ID as well as a method validation or a release or stability testing so that we can flex to whatever the client needs. And I think that's a really differentiating aspect of how we operate at our site.